Good evening, students. My name is Amrita Yadav, and I am your teacher. And I am taking the lecture of the Unit 6 and the chapter The Cell Culture and Genetic Manipulations. And in that, we have studied about that what is cell culture. This is class 12th biotechnology. And here we are reading the second part of this chapter, cell culture and genetic manipulation. We have discussed the basics already, like what are the important constituents of the medium, carbon sources, nitrogen sources, trace elements, growth factor, anti-foaming agents, energy sources, water. So these are the medium of the components of the medium. Cellular culture procedures, we have studied about the sterilization procedures that at 15 to 20 minutes, at 15 pounds psi pressure in the autoclave, the sterilization is done. And the medium also need to be sterilized. And before the sterile medium is added, the vessel also to be sterilized and the medium also to be purified, and it's made free of any other microbe before the starting of the culture and before the adding of the inoculum. Aeration and mixing should be done in the in the aeration tank. Large scale bioreactor, oxygen dispersed to achieve relatively uniform concentration. Many fermentation designs are there. And equipment of microbial culture, the baffle flask which is used in the laboratory, and the agitator and the shaker which are used in the water bath with the shaker are used in the laboratory. These we have discussed. So these are the laboratory procedures by which we can do the culture, and we culture the microbes. And this is the basic design of a bioreactor. This also we have studied about. And after this, studying about the types of the culture, that batch culture and the continuous culture, feed batch culture, and continuous culture are the three types of microbial culture. Continuous culture, feed batch culture, and batch culture. We have studied about the turbidity state and chemostate, that they are the continuous culture example. In chemostate, constant chemical environment is maintained, whereas in the turbidose state, the constant cell concentration is maintained. So these two are the important points, that what is the difference. The chemostate maintains the constant chemical environment, whereas the turbidose state, constant cell concentration is maintained. If the medium is fed to such a culture at a suitable rate, steady state is achieved eventually. At a steady state, the cell growth substrate consumption take place at a fixed rate. The growth of the cell remain constant during the steady state operation. This means that the steady state, the concentration of the cell metabolites and other things are balanced. So this is these are the difference of the culture and continuous culture and the batch culture. Microbial cultures are done by these methods. Measurements and kinetics of the microbial growth. A clear understanding of microbial growth is necessary for proper utilization of biological processes for production of metabolites. So clear understanding of microbial growth is necessary, and biological processes should be utilized in proper manner for the production of metabolites. In this section, the methods used for the measurement and quantitative evaluation of microbial growth will be discussed. There are 4 general patterns of microbial growth exemplified by bacteria, yeast, mold, and viruses. 4 general patterns of microbial growth are seen in these organisms. All these microorganisms grow in different ways. Bacteria grows by binary fission, yeast divide by budding, fungi divide by chain of elongation of branches, whereas the virus normally do not follow a regular growth pattern. They grow intercellularly in the host cell. Measurement of microbial growth. Microbial growth is defined as an elderly increase in an all chemical component in the presence of suitable medium and environment. So microbial growth ऑर्डरली इंक्रीज डिफाइन करी जाती है. इसमें सारे केमिकल कंपोनेंट्स होते हैं, सुटेबल मीडियम दिया जाता है किसी भी माइक्रोब को, वो ग्रो करता है. ड्यूरिंग द पीरियड ऑफ बैलेंस्ड ग्रोथ, डबलिंग ऑफ द बायोमास इज अकम्प्लीड बाय doubling of other measurable properties of the microbe such as protein, DNA, RNA, intracellular water. और बायोमास तो डबल होता ही है और दूसरी चीजें भी डबल होती हैं, जैसे कि प्रोटीन का लेवल, डीएनए का लेवल, आरएनए का, इंट्रासेलुलर वाटर का. इन जनरल मेजरमेंट ऑफ सेल मास और सेल नंबर इज यूज्ड फॉर क्वांटिटेटिव मेजरमेंट ऑफ ग्रोथ, सेल ग्रोथ. और इसके अंदर क्वांटिटेटिव मेजरमेंट भी की जाती है सेल ग्रोथ की. The parameter that characterize microbial growth is the doubling time. It is the time required for the cell to double itself. तो doubling time को ही ग्रोथ का पैरामीटर माना जाता है, क्योंकि टाइम लगता है सेल को डबल करने में ओरिजिनल वैल्यू के. और ये बैलेंस्ड फेज एक देखा जाता है ग्रोथ में, जो कि लॉग फेज होता है ऑर्गेनिज्म का, जब वो इक्वल अमाउंट में अपने आप को डबल कर रहा होता है. And the measurement of the cell mass or the number is one of the easiest way to measure the microbial growth. It is carried out by measuring the dry weight of the cell material in a fixed volume of the culture by removing the cell from the medium. So carried out ये कैसे करी जाती है ड्राई वेट मेजर करके सेल मटेरियल का फिक्स्ड वॉल्यूम में ऑफ द कल्चर बाय रिमूविंग द सेल्स फ्रॉम द मीडियम. मीडियम में से सेल्स रिमूव किए जाते हैं और उन्हें ड्राई किया जाता है टिल कांस्टेंट वेट इज ऑब्टेन, जब तक कांस्टेंट वेट ऑब्टेन नहीं होता. फॉर ई कोलाई, द बैक्टीरियम दैट एक्सटेंसिवली यूज्ड इन लेबोरेटरी, द ड्राई सेल वेट इज 1 बिलियन सेल्स इज अप्रोक्सीमेटली 150 मिलीग्राम. 1 बिलियन सेल का वेट 150 मिलीग्राम होती है. यानी 150 मिलीग्राम में 1 बिलियन ई कोलाई के सेल आते हैं. सेल ग्रोथ को मेजर करने का और भी तरीका है एब्जॉर्बेंस कैलकुलेट करना सेल सस्पेंशंस की स्पेक्ट्रोफोटोमीटर में. तो अगर हम एब्जॉर्बेंस कैलकुलेट करते हैं सेल सस्पेंशन की, एक डिवाइस आती है स्पेक्ट्रोफोटोमीटर, उसके थ्रू, तो हमें ये प्रिंसिपल मिलता है, ये फैक्ट मिलता है कि छोटे मॉलिक्यूल्स स्कैटर करते हैं लाइट को प्रोपोर्शनेट टू देयर कंसंट्रेशन से प्रोपोर्शनेट अमाउंट में लाइट को स्कैटर करते हैं. व्हेन लाइट पासेस थ्रू अ सस्पेंशन ऑफ द बैक्टीरिया, देयर इज अ रिडक्शन इन द लाइट ट्रांसमिटेड एज अ कॉन्सिक्वेंस ऑफ स्कैटरिंग. जब लाइट पास होती है सस्पेंशन की बैक्टीरिया की, देयर इज अ रिडक्शन इन द लाइट ट्रांसमिटेड एज अ सीक्वेंस ऑफ स्कैटरिंग. सीक्वेंस ऑफ स्कैटरिंग जो होती है, लाइट ट्रांसमिट होती है. thus with the different cell concentration, the absorbance of a particular wavelength will be proportional to the concentration. The cell concentration of the unknown microbial sample can be calculated. और सेल की कंसंट्रेशन जो है वो एब्जॉर्बेंस जो है वो प्लॉटेड करी जा सकती है सेल की कंसंट्रेशन से. और सेल कंसंट्रेशन अननोन माइक्रोबियल सैंपल की मिल जाती है, can be calculated by measuring the absorbance of the same wavelength. Apart from the method described above, other methods are also used for measuring cell growth. अपार्ट ये मेथड तो यूज होते हैं, अदर मेथड जो बाकी मेथड जो होते हैं वो सेल ग्रोथ को मेजर करने के लिए वो क्या होते हैं, मेजरमेंट ऑफ वेट वेट ऑफ द सेल्स, स्टेबिलिटी मेजरमेंट, एटीपी मेजरमेंट, वायबल कॉलोनी फॉर्मिंग यूनिट्स, वायबल प्लेट काउंट भी इन्हें कहते हैं, कॉलोनी फॉर्मिंग यूनिट्स भी काउंट करी जाती हैं. और एंड यूज ऑफ कोल्टर काउंटर नाम का इलेक्ट्रॉनिक इंस्ट्रूमेंट भी यूज किया जाता है फॉर डायरेक्ट काउंटिंग ऑफ माइक्रोबियल सेल इन सस्पेंशन में कितने सेल्स हैं. In Coulter counter, the cells are made to pass in a single file through electrical field and the electrical impedance thus generated is recorded. जितनी इलेक्ट्रिकल इम्पीडेंस बनती है वो जनरेट करी जाती है. कोल्टर काउंटर के थ्रू सेल को काउंट किया जाता है और सिंगल फाइल के थ्रू सेल्स पास किए जाते हैं इलेक्ट्रिकल फील्ड में. ग्रोथ काइनेटिक एंड स्पेसिफिक ग्रोथ रेट. इन ऑर्डर टू अंडरस्टैंड microbial growth kinetics, we shall take bacterial binary fission as an example and each cell division produces two identical daughter cell. Growth kinetics हमें पता ही है, हर cell divide होकर टू आइडेंटिकल डॉटर सेल बनाता है. इसे जनरेशन जितना टाइम ये लेता है दो डॉटर सेल बनाने को, उसे जनरेशन टाइम कहते हैं. जनरेशन टाइम इज ऑल्सो नोन एज डबलिंग टाइम, डीडी, टीडी बोला जाता है. And because the population of cell doubles over this time. For example, one cell of the E. coli put in nutritive medium will divide every 20 minutes. After 1 hour of growth, that is 3 generations, one cell will be becoming two say 4 say 8, because cell number double हो रहा है हर division में. Increase in cell number over the time is exponential और logarithmic. तो ये logarithmic रहता है increase, क्योंकि cell double होता है डबलिंग टाइम में. तो एक्सपोनेंशियल ग्रोथ ये रहती है. because and a typical bacterium growth curve is depicted in figure, and about which you have already learnt in unit 1 of the textbook for class 11th. In the lag phase, the microbial population remains almost constant. It is, however, a period of intense metabolic activities as the bacteria adapt to the culture condition. आप एक इंटेंस मेटाबॉलिक एक्टिविटी के बाद बैक्टीरिया कुछ कल्चर कंडीशन में अडेप्ट करता है. वंस द सेल हैव अडॉप्टेड टू द कल्चर, सेल डिवीजन अकर्स एंड इंक्रीज फ्रीक्वेंसी until the maximum growth reached. This is called log phase. At this point, exponential growth occurs and the cell biomass or the cell number increases as constant rate. यहाँ पर इक्वेशन भी दे रखी है किस तरह से ग्रोथ रेट की इक्वेशन होती है. So that also we can understand. Specific growth rate mu is equal to 1 upon x dx dt. The specific growth rate is an index of the growth of rate of the cells in a particular environment. In other words, the specific growth rate is characteristics of microorganism and is function of the growth environment, including temperature, pH, medium composition, and the level of dissolved oxygen. During the period of true exponential growth, equation 1 can be integrated to provide the following equation. Xt is equal to x0 e mu t. Xt is the biomass concentration after time t. X0 is the biomass concentration at the start of the exponential growth. E is the base of natural logarithm. Taking natural logarithm, ln is equal xt is equal to ln x0 plus mu t. This equation is the form of the straight line y is equal to mx plus c. Thus, for an exponential growing culture, a plot of natural log of the biomass concentration against time, the semi-log plot will give rise straight line with the slope equal to mu. Mu is equal to ln xt minus ln x0 by t. Please note that mu is equal to 2.303, that bracket log xt minus x0 upon t. We consider a situation where the time is 0. At time 0, the cell biomass x0, then after exponential growth is equal to 1 doubling time, xt is equal to 2x0. When t is equal to td, 2x0 is equal to x0 e mu td. And taking natural logarithm, it comes td is equal to log 2 upon mu. So this will come, doubling time is equal to log 2 value upon mu. So 0.693 upon mu is the doubling time. So let us now second approach, which examine the relation of the cell to the cell number, where number of the cells are given, number of the cells and the start of the exponential growth is given by n0. Number of cell division, when it is 0, then the number of cells are 1 and mathematically is represented n0. Number of cell division is 2, then the number of cells are 2, and n0 into 2, that is n0 to the power 1, that is the number of division. Here the second division is occurring, so m0 to the power 2, that means 4 cells will come. Here when the third division is occurring, that means n is equal to 3. So 2 to the power n0 into 2 to the power 3, that is 8 cells will be there. n0 into 2 to the power n is the formula for the number of cells in the nth division will be given by nth division पे कितने cells होंगे वो इस फॉर्मूले से पता चल जाएगा. जब n नंबर का डिवीजन होगा तो n0 जो सेल्स थे इनिशियली इंटू 2 to the power n उतने सेल्स हमें मिलेंगे. nt is equal to n0 2 to the power n. एंड टेकिंग लॉगरिथम, जब हम इसका लॉगरिथम लेंगे तो log nt is equal to log n0 plus n log 2, and thus the number of division n have taken place. Then n is equal to log nt minus n0 upon log 2, and log nt minus n0 log nt minus n0 upon log 2 की वैल्यू 0.301, and that will come 3.3 log nt minus n0, and total growth time upon number of division t by n will give you doubling time. And when you will there is one numerical is also given. First, calculate the generation time doubling time of a bacterium population in which the number of bacteria increases from 10 to the power 4 to 10 to the power 7 ml during 4 hours of exponential growth. First, calculate the number of division the population must have undergone to increase from 10 to the power 4 to 10 to the power 7 in 4 hours. So using the equation 9, n is equal to 3.3 10 raised power 7 minus log 10 raised power 4 is equal to 3.3 10 minus 4 is equal to 10. TD is equal to 240 minutes per 124 minutes. That is scale up of microbial process. That this is the log equation. Then how we do scale up, that is from small culture flask or small laboratory scale bioreactor, how we will be, or we can say pilot plant. From pilot plant, how we are going to scale up our bioreactor conversions into large industrial biofermenter. That is meaning is the scale up. हम कैसे हाई कंसंट्रेशन में चेंज करते हैं अपने छोटे बैच कल्चर को, इसको स्केल अप कहते हैं, स्केलिंग अप इसको कहते हैं. तो दैट वी आर गोइंग टू अंडरस्टैंड इन द नेक्स्ट वीडियो. Tomorrow I am going to explain you about the scale up. So with this, I hope you have understood about the growth curve and the growth kinetics, which I have discussed with you. So with this, I would like to end the video here. So like and subscribe my channel, and for more such biotechnology video, share with your friends who are reading biotechnology, and happy learning. Thank you and have a nice day.